We're now going to consider the diversity of organisms, and specifically we're going to consider how the diversity of organisms is classified, and then we're going to consider interpretation of this diversity in an evolutionary context. So if you look at the pictures on the screen here, you can see that it will remind you that organisms come in a variety of different shapes and sizes and types, and some are terrestrial, some are aquatic, some are vertebrates, some are plants. How is it that that organisms are in fact classified? And humans do tend to notice similarities and differences, and we classify everything and group everything. Just consider, for example, going into a grocery store. You are not surprised when you see all the canned goods together or all the cereals together. In fact, even within the canned goods, you see different groupings within the canned goods, such as different kinds of canned beans tend to be near each other on the shelves, or canned fruits tend to be near each other on the shelves. We do notice similarities and differences and classify based on these shared similarities or differences. And this is true as well for living organisms. But before we consider classification, though, let's remind ourselves of what organisms actually are and what some of the attributes of organisms are. We can recognize an organism because an organism is made of cells. An organism might only be made of one cell, meaning that it's unicellular, has one cell, or an organism can be made up of a large number of cells, such as all of us. And so where do those cells come from? Well, a cell is made by another cell, right? You learned in your previous classes that cells produce more cells by cell division. So a cell comes from a cell via cell division. Well, where did those cells come from? Those came from other cells by cell division. Well, where did those cells come from? Those cells were made by cell division, right? We can go back and forth, back and back and back, earlier and earlier and earlier and earlier. Cells made by cells made by cells. And you can extend this, not completely indefinitely, of course, because if we go far enough back in time, all of a sudden we hit what? We hit the genesis of the solar system, and we hit the Big Bang, and so on. So obviously there was a starting point. At some point, then we hit, where did the cells actually start? And we'll return to that important topic later. But for right now, let's just consider that organisms are made of cells, and that these cells are made by other cells. Another common characteristic shared by all organisms is that we all share the same genetic material, meaning double-stranded DNA. You learned a lot about this in your earlier biological classes. And individual organisms are generated by other organisms, and specifically by organisms of the same type or the same species. There are rare exceptions to this, for example, hybrids, and we'll be talking about this when we talk about definitions of species and the process of speciation, but generally individual organisms are generated by other organisms of the same species. And so we can run through the same thought process that we just did with cells. So an organism was generated by organisms of the same type. Those organisms were generated by organisms of the same type. Those organisms were generated by organisms of the same type too. And as we go back and back and back in time then, we might expect to see differences. So we'll return to that point in just a second. But first, let's pause for a question to consider if we really understand what an organism is. So let's compare, for example, the difference between an organism and a virus. So I'd like you to consider this question. It's like a pseudo-clicker question. So the question is, an organism is different from a virus, and an organism blank, while a virus blank. Now, I've given you 3 different possibilities to consider, and I would like you to pick what you think is the best answer for this question. The way to consider this is that the part of the answer before the comma, you would put in the first blank, and the part after the comma, you would put in the second blank. So, for example, if your favorite answer was A, then you would complete the sentence by an organism is different from a virus, and an organism always has double-stranded DNA, while a virus never has double-stranded DNA. We would pick B if you think it's an organism that's different from a virus, and an organism is made of cells, while a virus is never made of cells. Or if you want to pick C, then this would be completed by an organism is different from a virus, and an organism always has double-stranded DNA, while a virus does not have genetic material. It just uses the hosts. So please pause the movie for a second while you consider your favorite answer. Welcome back. Hopefully you picked B as the correct answer. In fact, viruses are not considered organisms because they're never made of cells. And we'll come back to viruses later on in the course. But let's return now to the topic of how are classification systems used to identify similarities and differences between different living organisms. The first point I'd like to make is that classification schemes are hierarchical, meaning that they are nested or that they consider subsets of groups. And the classification systems that we use today have evolved over hundreds of years. One of the individuals who is most closely identified with generating some of the features of classification schemes that are still used today is Carolus Linnaeus, a very influential European biologist of the 1700s, who recognized that there were huge differences between different organisms that would be considered animals versus different organisms that would be considered plants. And he described many different organisms and described them as being more suited for the so-called plant kingdom or the so-called animal kingdom, meaning that they shared attributes with other animals or other plants that were completely distinct. So these then would be different subsets of all living organisms. Now, another thing about hierarchical classification systems is that the different taxonomic units, such as a kingdom, so the animal kingdom, for example, would include all animals. And then within that kingdom, there would be a number of different subgroups. The name for the different subgroup of the next level is phyla. Phyla is plural, phylum is singular. So there are a number of different phyla in a kingdom. These can then be subdivided into classes, which can be further subdivided into orders, which can be further subdivided into families, and so on. First of all, you do not have to memorize all these different subgroupings and exactly where they are in the hierarchical scheme. But you should at least recognize these names so that you do understand that a family or order or a class is in fact a taxonomic unit that has a specific meaning and a place in the hierarchical scheme of classification, so that if one is considering a family of a specific group of animals or plants, for example, then it should make sense to you that it would be one of many families, for example, clustered together in an order, and that members of a family are grouped together because they share certain characteristics with other members of the family. So that's what's meant by hierarchical. There are different levels of inclusiveness or exclusiveness. These are nested in that they are subsets within more inclusive, higher-level taxonomic groupings. Another very important thing that Linnaeus did is that he established a convention for naming species that is still used today. And this was in contrast to the conventions of the 1600s, which was to use very long descriptions for a species, and he shortened it down to just a 2-word name for a given species. And we still do this. So, for example, our own species is species Homo sapiens. Species always have 2 words. The first word is, in fact, the genus. So if we go back here, the genus is the next level up, a taxonomic level up from species. So a genus typically includes a number of different species. Sapiens is in the species within genus Homo, and the full proper name for our species is Homo sapiens. So there is 2 words. It is usually italicized or underlined. That's the convention. The genus name is always italicized, and the species name is never. It is always capitalized, and the species name is never capitalized. Those are the conventions that we expect you to follow, and is followed by your textbook. Now, over time, as biologists learn more about different kinds of organisms, they realized that there were other organisms that didn't quite fall nicely into one of the 2 kingdoms. So, for example, there were many protozoans that we'll learn about later that shared some characteristics with animals and other characteristics with plants. They didn't seem to either be very clearly animals or plants, and so a new kingdom for the protozoans was in fact generated later in the 1800s. Bacteria as a separate kingdom was generated in the 1930s. Fungi were added even later. And so by the time that the 1960s rolled around, there were 5 different kingdoms: animals and plants, protozoans and bacteria and fungi, identifying these groups of living organisms as being completely distinct from each other. And yet within one of those kingdoms, they shared characteristics which were not shared by individuals in the other groupings. And so that was the classification system. So these classification schemes then are based on shared characteristics. An important conceptual breakthrough then was provided in the 1800s when it was recognized that these shared characteristics reflected evolutionary relationships or degrees of relatedness. That more closely related organisms are more similar to each other, and this is not only true within a species, but also between species or between larger taxonomic levels. So then the classification schemes are not just a description of the shared characteristics, but are also a description of the evolutionary history of living organisms. Now, when scientists were very interested in trying to look at the relationships between these different kingdoms, then it's fairly difficult to try to understand when things are very disparate from each other. Imagine an animal or a plant or some of the microbes or fungus. How do you pull out shared characteristics between all these different groups when they're so different and distinct from each other? And the answer, of course, lies in molecular characters. So instead of considering characteristics such as presence or absence of hair or mammary glands or aspects of bone formation, then with the advent of different kinds of molecular techniques, it was possible to use molecular characters to look at shared characteristics. And a tremendous breakthrough of understanding the relationships of the major groups of living organisms came from a set of studies done by Carl Woese and many of his colleagues, in which they studied a molecular characteristics which were could be used for all living organisms, because they used a specific small subunit of ribosomal RNA, which was found in every single organism, from bacteria to the plants to the fungi to the animals. And this is something that could be used to compare all groups of organisms. So a quick reminder here, and this is figure 1.4 from your textbook. In ribosomal RNA, then, we have a linear sequence of a number of different ribonucleotides. And so we can show this diagrammatically using the single letter abbreviations for the 4 different types of ribonucleotides. And so one can look at the pattern of, or the sequence of ribonucleotides. And so, for example, in this short section of a much longer sequence, then one can compare and contrast. Here are 8 ribonucleotides shown. You can notice that the first 4 are the same, and then they differ in this one location here. And then there are 3 more that are, again, identical. And so, for example, if all land plants have a C at this same location where the green algae have a G, then this is a distinct characteristic that allows one to separate these 2 groups, where the land plants all have a C at this location, green algae have a G at this location, and they share all these other attributes. So it's a combination then of the shared and the different characteristics that allows one to classify and understand how organisms are similar and then how they are different. So the logic then, in relating these molecular characteristics back to their relatedness, is that more closely related species should have ribosomal RNA species that are more similar to each other than ones from more distantly related species, because they share a more recent common ancestor. So the way of showing these relationships then is to show this diagrammatically using something called a phylogenetic tree. Now, some of you may be familiar with this depiction, and some of you may not. But this is a phylogenetic tree representation of the ribosomal RNA results from the study that was just being described. And this was a huge surprise to the biologists who are used to thinking in terms of the 5 kingdoms. And so imagine if one understands that the living organisms, or if one assumes that living organisms can be separated logically into 5 extremely distinct groups, and one would expect 5 major branches to fall out of a phylogenetic tree that was generated from the results. So here's the phylogenetic tree right here, and the different taxa are shown here on the tips of this phylogenetic tree, and they're grouped or clustered together based on shared similarities. And the first thing that one can notice is that the 5 traditional kingdoms do absolutely not fall out cleanly on this phylogenetic representation at all. In fact, here's the animal kingdom, the fungal kingdom, and the plant kingdom are just 3 tips buried within this huge domain, which includes all eukaryotic organisms. And in fact, there are 3 major and fairly distinct groups of living organisms, which include the bacteria, another group of prokaryotes called the archaeans, and then all the eukaryotes then come together. So the 5 kingdom classification system does absolutely not adequately describe the true diversity of organisms that is revealed when one examines molecular characteristics. And so while some of these names are still useful, we still talk about fungi and animals and plants and so on. The diversity of living organisms is no longer identified as being adequately represented by this 5 kingdom classification system. And now the emphasis is on describing or recognizing the huge differences between the 3 domains, and then within the different domains, understanding the differences between the major lineages within these domains to understand the characteristics of the diversity of living organisms on our planet.